Hi, everyone. Hello, and welcome to today's webinar where we are covering evaluating needs for biocompatibility testing and mitigating risks when changing your medical device. It's a mouthful, but we're going to make it fun today. Thank you so much for taking time out of your afternoon or morning or evening to join us. My name is Taylor Brown, and I am a senior medical device guru here at Greenlight Guru, and I'll be your moderator for today's event. We have a really special presentation for you. I know our presenter, Thor Rollins, is really looking forward to sharing his expertise on the topic of biocompatibility testing, as well as risk mitigation practices, specifically in the context of making changes to your medical device. And before we dive in to the presentation from Thor and Nelson Labs, I'm going to touch on a few items. Today's webinar will run for about 75 minutes and it will include a Q&A session at the end. Thor's already told me that we'd like to focus on your questions, so feel free to submit them as they come up. Again, we'll save some time and hopefully it's a lot of time at the end for your specific queries. Yes, we are recording. Yes, slides will be made available. So look for an email from us later today. While I'm up here, I'd like to share a few words about Greenlight Guru and why we put on these free training sessions. If you've been to one of these sessions before, then you already know we put these sessions on because improving the quality of life is our mission here at Greenlight, likely a similar mission as many of you listening today. Whatever we can do to fulfill that mission, we will, whether it's through hosting these free sessions or creating an entirely reimagined training experience altogether. With Greenlight Guru Academy, you might be wondering, what is Greenlight Guru Academy? Well, it is a medical device specific educational platform built from our library of content resources and taught by a few subject matter experts, gurus like myself, and many others on my team. We'll frequently be adding new courses and learning paths on design control and risk and regulatory submissions, biocompatibility. So after today's presentation, visit academy.greenlight.guru to enjoy. If you need an access code, you can use explore_engage_educate. If you have any questions about what I just rattled off at you, let us know and we can send you those details as well. So in addition to our training programs, we fulfill our mission by partnering with world-class medical device consultants like today's presenter, Thor Rollins, as well as through our award-winning medical device QMS software. If you're interested in said software, visit our website at www.greenlight.guru and schedule your free personalized one-on-one demo. So enough about me and Greenlight. Let's get on to what you've all been waiting for. Let me give Thor a proper introduction here. Thor Rollins is a certified microbiologist and specializes in the selection and conduct of in vitro and in vivo biocompatibility tests. He actively speaks on biocompatibility related topics through Nelson Labs external seminars, webinars, and trade shows. And we talked about before this webinar started, we're excited to both get back on the road and see you all in person later on this year at many different conferences. In 2013, he presented on biocompatibility at the American College of Toxicology and has published many articles on the matter. He is a participating member of ISO committees and AAMI committees and plays an active role alongside the FDA to develop standards, discuss biocompatibility methods, and everything in between. So without further ado, let me hand it over to your presenter, Thor Rollins.
Thanks, Taylor. And I really enjoy these conversations. For some of you who may have participated in some of my webinars and other things that we put on in the past, I do have to agree with Taylor. I am having a little bit of webinar burnout like everybody else. I'm really missing seeing people's faces when I talk, other than my own, which is the scary one. So I'm hoping that someday soon we'll be able to do this in person and see everybody. So thank you, Taylor, for the introduction, and let's go ahead and jump into today's message and training. Now, just to emphasize what Taylor said, I do have content I'll go through, and I'm going to talk about what we see right now today about changing medical devices and the activities that are needed to justify those changes. But part of this particular conversation is the questions, because you guys might have different circumstances or different areas of changes that you want to talk about. And I've been doing this for about 20 years now. And so we see quite a bit of different changes in medical devices. So I'd really like to give an ample amount of time to address questions or situations, I think everybody would be kind of it'd be useful. So go through the slides and the basic ideas behind that, but really want to emphasize the questions coming in. So let's get into it. The testing and risk management of changes of medical devices, especially when you look at biocompatibility. So the first thing that you start thinking about changes is what constitutes a change, and this is probably most of the time when I'm dealing with changes, I'm dealing with engineers, engineers that are developing or supply or getting supplies for these medical devices, and some of them want to kind of really understand what is a change that's impactful for biocompatibility, and maybe what are changes that are not. That's pretty much the most valid question to talk about. So if you think about it, first off, is it an impactful change? And if it is an impactful change, how impactful is it from a risk-based approach? And that basically determines the approach for testing from the low scale to the high scale, right? So that's what we're going to go through today. So when you first start looking at it, what constitutes a change? The first thing that I always want to point out here is, why are you changing? I always joke, but it's actually probably not much of a joke, but engineers just love to fiddle with things. They just love to change. And yes, I think we all want to make things better and more efficient and more safe and things like that. And those are great changes. But sometimes we're just changing because it's time to put out a new version of the device. And so we're changing a little bit here and there. Those are a little bit harder to come from a risk-benefit ratio. Other times we're changing because the supplier no longer supplies that material. And so we're going to have to either resource another material, or maybe they have a one that they're calling, the same, which I hate that word, that's the same, because that's kind of a ambiguous term when it comes to materials. So the first thing you have to really think is, why are you changing? And I always push back a little bit because once you have an approved device and you know that device is on the market, it's safe. If you're just changing for the sake of change, that's tough. But if you're changing to improve the benefit to the patient, if you're doing it out of necessity, those kind of things, obviously those are worthwhile exercises and worth the time and money that goes into justifying these changes. So the first question is, why are you changing? And I do want to say that any change to a medical device, be it the packaging, being the sterilization procedure, being it the processing, the additives into the polymers, anything that's being changed should go through a biocompatibility evaluation. That doesn't mean you have to repeat testing on any change. That's not what I'm saying. I'm saying the evaluation needs to be done. Lots of times in my past, we've seen changes of packaging, for example, and the individual company decided not to do any testing because they determined that the packaging had no impact on the biocompatibility of the device. We know that packaging leaches chemistry that could be transferred to the device. We've seen ink that's been leached off of a label that has been interacted with the medical device. So there are times where you may not consider the change to be even one to think about and just pass over, and that's the risk. I really want to put hammer home that any change should go through an evaluation, and this doesn't mean evaluation from expert consultants from our end. It means internally looking at that from a biocompatibility perspective, maybe doing some high-level evaluations, just trying to understand, is there a risk for this change? And we'll kind of go through some of those that are some of the concepts that go into those evaluations, so you can do those internally. Now, if you're changing your device with the intent to significantly affect the safety or effectiveness of the device, that's where the FDA says more in-depth evaluation may be needed, including a 510K. And so, there is a difference between the evaluation that's needed to evaluate safety and maybe not needing a new 510K for an FDA or a new CE mark, for example, or something like that. And there's another change that impacts the regulatory status of your device. What I'm talking about today is more about just evaluating the safety. And that could be done on something that doesn't need a new 510K, but you still need to evaluate the impact of that change, or one that does need a new 510K based on the change. And obviously then you need to have a little bit more ducks in your row, because the FDA is probably going to be closely evaluating your documentation and your rationales with those changes. So here are some of those things that we see routinely, like So we do, every day, we do biological evaluation plans, we do gap analysis, we do failure investigations. My team is involved every day in these things. One of those things we do almost, like I said, pretty much all the time, every day we get calls in for this, is changes. We see it every single day where a change happens. Some of these changes are, like I said, label changes. This could be what you're actually saying on your label, like pyrogenicity claims, latex claims, things like that. Obviously, that we have to evaluate those claims. But this also could be the label itself. We talked about at the beginning that we've seen leaching from labels interact with devices. So if you're putting a lot of black ink, for example, in your label, and you have humidity in your sterilization, then there's a possibility of leaching from that label. And so we have to just evaluate it. Now, if there's multiple barriers, that's kind of things we would look at, but that's the part of the assessment that needs to be done. We talked about packaging kind of already. Some people don't even think about adding a plastic insert into the device and how that could have an impact on your biocompatibility. Like I said, if you're having claims of non-plasticizer on your device and you're adding something into it that could have some plasticizers, DHP or PPA, then obviously that impacts not just your safety, but your label claims. So things to think about. The most common, though, we see are the materials and process change. Either you're changing a complete process, a cleaning process, for example, or you're changing a fluid during the process, like a mold release or a milling compound, or the material, you're improving the material, you're changing durometers of a plastic. All these things we have to evaluate from a risk-based approach. The one that drives me crazy is adding colorants. We had a manufacturer not too long ago that, and this isn't a joke, I wish it was, they actually had a surgeon reach out to them and said, hey, I really like the color red. If you could change your device's handle to red, I think that would, maybe I'd buy more. I think I'd choose your device over your competitor's device because, red, I'm not sure the reason that the surgeon just asked for it to be red. So they did. They wanted to look at including that colorant into the handle. If you want to know the possibility of changing colorants, the FDA did a whole webinar on colorants in medical devices. I also have on these slides, which I will make available to everyone afterwards, the white paper and a webinar that I did on changing colorants. Just be careful when you change colorants. It's one of those that there are times when changing colorants it's a good thing. Or adding a colorant is a good thing, I should say. Like if you're matching sizes and it's easy for the surgeon to match colors instead of trying to figure out the sizes, those things have their place. But for the most part, if you're adding a colorant to a patient, either direct or indirect patient contacting portion of the device, that's adding something that could be fairly toxic depending on the colorant, and there's not really a benefit to the patient. Unfortunately, having it look pretty really doesn't help the patient out, right? So those are things to think about. We also look at location changes. It's been a few years now, but we had a manufacturer that moved their manufacturing just a mile and I think mile and a half, just barely over a mile down the same road. So really the address changed, and they picked up all their equipment from their processing facility, all the same personnel, and just moved a mile down the road to a bigger location. And they called me up and said, hey, do we even need to do biocompatibility? We're not changing the process. We're not changing the material supplies. We're just changing locations. And I said, that should be a pretty easy evaluation. And we recommended doing a cytotoxicity test. Well, maybe surprising, maybe not. The new manufactured device failed cytotoxicity and the old one passed, right? So they did a comparison. And so they did some chemistry to evaluate the reasoning, the difference between those two manufactured parts. And we found one chemical in the new part that wasn't in the old part. And when we did some toxicological research, we found out that it was a chemical that was used in manufacturing surfboards. And when we told them that, they just happened to say, our new neighbor manufactures surfboards. And this is a very volatile compound. It got up into somehow got up into the ventilation system and got into their mix into high enough levels that it felt cytotox. Now, I probably would never have guessed that, and we've dealt with moves tons of times, and we've never seen that before. But that just goes to show, or sense, I should say, but that just goes to show you that there are changes that could impact biocompatibility that could be surprising. right? So just things to think about when you're looking at changing in biocompatibility. So when you're looking at this, though, any change, like I said, should be assessed, not necessarily tested. Though I want to make sure everyone's aware of that, because I don't want you guys to think that I'm telling you you have to test on every single change. Every single change needs to be evaluated based on the risk to the patient. And that first step is always initial risk assessment. And so the first thing, and these are some of the areas that we want to look into when we look at risk assessing for biocompatibility. Where is the change at in the device? Are we contacting, patient, are we changing patient contacting materials? Are we changing, that could be direct or indirect, right? So, the question always comes up, well, if it's in the handle, that's sterilized together. There could be humidity present. There could be leachables that come through. And those are all concerns we want to evaluate. So I'm not saying that if it's in the handle that you're changing, that you're automatically clear. You still want to understand the materials that you're changing to, the likelihood of that leachability happening from your handle to your patient contacting portions. But those are things that go into the risk assessment. The other thing to think about is, are you changing a portion of the device that only contacts intact skin? And if you are, the FDA has a new draft. I have to put out the word draft because we had some, responses from the FDA that they're not official yet, but the thought process is still there from a risk-based approach. There's a new draft from the FDA about skin contacting materials. So if your change is still working within that same kind of thought process of an intact skin contacting and using those materials, I did a webinar not too long ago called on that draft document called the Naughty and Nice list. So you can look that up. But those are things you can put into your risk assessment. Is the portion of the device that's changing where it contacts the body. Then once again, the intent, if you're significantly increasing the safety of the device, then that helps with your risk-benefit ratio. If you're increasing the benefit of the device to the patient, that helps with evaluating the risk. I'm not saying that you can just say, oh, it's safer now, we don't have to do any testing or any evaluations. That just helps in evaluations to understand if you're making this device much safer, that helps with your risk evaluation. Is it to mitigate a known risk? Based on clinical data, do you know if your device is having issues and you're changing that to improve it? That goes in the same kind of idea that we just talked about. Are there any unintended consequences of these changes? Are you seeing things happening based off, this is why you do the evaluation. We talked about the packaging, we talked about the moving, things like that. So that's what you want to cover, is how do you screen for unintended impacts? And then ultimately, the end of this risk assessment is, do you need to test? Do you feel confident that you can justify that the testing is not needed? Or do you feel like you have to do some testing to help justify this? So these are the things we put in the risk assessment when we do it for a change, but obviously you can do these internally too. So I just want to throw up this slide. This is one of my favorites. I actually stole this from, well, I borrowed because he gave me permission, from Ed Rivardi. He works at J&J. He let me use this slide. I think it's a great quick capture of what risk is, right? So that little mouse is looking at that cheese and trying to look at the benefit risk ratio, and maybe the helmet helps mitigate that risk. So, that's really the whole part of this risk assessment when we look at changes, is the combination of the probability of occurrence of harm and the severity of that harm. So those are the inputs we put into those risk assessments to understand the possible impact of the change. And it's always about the risks that you don't know. And that's where you have to try to cover yourself, those unintended ones like I talked about. So we actually used the FDA not draft this time, this is actually a guidance document. So it was released in 2016, updated in 2020. Let me say reissued in 2020. There wasn't a whole lot of updating, but it's a risk management of biocompatibility. I'm sure if you're on this webinar, you guys are very familiar with this document. But this paragraph in this document is my favorite paragraph in the document, which just explains the risk-based approach, which is you look at the material components, the manufacturing processes, the clinical use. So those are your three inputs into any risk assessment for biocompatibility. You consider those information, and then potential risks from the biocompatibility perspective are identified and then mitigated, either by testing or evaluations. So this is from the FDA. This is the Bible which we live by when we do risk assessments. So when you're looking at your changes, you still follow those formulas. You look at the materials. Have you changed any of the materials, right, with your change? And this is patient contacting either directly or indirectly materials. Have you changed any processes that could impact those materials? Have you changed the intended use of your device, which sometimes they add another indication, right? So we have to look at that gap. So those three things, inputs, go into your risk assessment. And based on that, you can either look at the risks and then identify if there's testing needed or if we feel confident in assessment. So initial thoughts for your change, like we talked about, patient contact material, direct or indirect, material similarities. Have you used these materials previously on another device? Are you using the same supplier and gone on the same processing of another device? Now you're just adding it to this device. We'll kind of talk about some considerations with material similarities in a minute, but this is a good way to kind of help, make sure your testing is under control. If you can use materials that you can show to the FDA or even to yourselves that you know a lot about and you're safe to use. And then the last one is patient contacting surface area. And this is the one that gets some people kind of caught up. So it has to do with kind of dose from a toxicological standpoint. Toxicology 101, it's all about dose to the patient. Everything is toxic and everything is non-toxic. It just depends on how much of it is given to the patient. And so you can have a device that you've tested biocompatibility, you've done extractables and leachables on, it has tox assessments, you've gone the whole way, and it's approved on the market. And now you're thinking about adding length to that device, or maybe you are increasing the colorants that you already have on there, or maybe you're now adding more of a coating or something along those lines. Remember, it's about dose. And so even though you're using the same materials, same processing, things like that, you're now increasing the dose of a certain material. We have to evaluate that as a potential risk. Now, if you've done chemistry, we can kind of look at those margins of safety and the tox risk assessment to understand how far away some of the toxicity endpoints may be. We might want to look at the surface area changes. Is it within a certain ratio? Is it greater than that? Those are considerations, but I've seen a lot of people increase the length of a catheter, for example, and then decide that their testing is completely great. A little side thoughts here, and it has more to do with the initial biocompatibility than it does anything. Biocompatibility is done on surface area to volume ratios. The traditional biocompatibility is, at least. And so when you do that, make sure you work with your lab to really make sure you're testing the ratios that have the final device, right? Or the worst case possibly if you're doing a family of devices, to make sure your biocomp is true to those surface area ratios. Same concept. If you're testing only the same surface area of different materials in like a coupon or a family device, but the worst case one has more of a certain material, we want to make sure that we test the right ratio, the worst case ratio. So just make sure you work with your lab or your biocomp experts when you're doing initial testing on something like that to make sure your surface area ratios are good, because it plays in the case here with the change. Okay, so the other thing to look for is, especially when you're changing materials, is the information you can get from your suppliers. This is essential. I talk to suppliers a lot, especially polymer suppliers, and I try to convince some of the amount of testing that they could put into a supply of a plastic to help sell their device, right? Or their materials. So if I'm a medical device manufacturer and I'm looking to add or change a material to my device, if I have all the testing already done with a sterilized device, how I'm sterilizing my device, that risk assessment becomes a lot easier compared to something that has maybe just cytotox or maybe even a USB class 6. Those don't really mean a ton when it comes to looking at some of their endpoints. So if you're doing a big change and you have to evaluate sensitization, for example, or implantation, for example, or things like this, cytotoxicity and even a standard USB class 6 aren't really going to help you in evaluating all those endpoints. So take a time to really talk to your suppliers, look at agreements, right? Try to get them not to change. I just got off a phone call this morning with a company that we have to repeat a lot of biocomp because they changed the catalyst in their material. Well, if they could, and I know they don't have a lot of control over their suppliers, but obviously that their supplier didn't change that catalyst, things would have been a lot better off for their biocompatibility. So just keep that in mind. Composition disclosures, I love this. You're not always going to be able to get these from your suppliers, but If your supplier can get you exactly what's in their material and exactly the percentage of those, then we can look at that from a toxicological standpoint and even see if this change has any kind of impact from a safety standpoint. You also have to think about their processing. Could they leave something on the device, on the materials that could impact your biocompatibility? So you have to get a good sense of their processing and also their cleaning, if they clean, hopefully they do, right? You can always get MSDSs or MS, material safety data sheets, but those don't really help, honestly, a whole lot. I hate to say that because everybody throws us over an MSDS when they're asking us to evaluate a material. They're not peer reviewed, so we even see mistakes in those. And they may have like irritation, maybe, but they're not great. And they're not really a lot of times on the process device. It's just on the raw material. So it kind of depends on the type of material you're getting from a supplier. So those are not my favorite. Other versions of that, though, is something called the device master file. We see these for a lot of really good suppliers where they will do a lot of biocompatibility testing, sometimes all the biocompatibility testing. Some suppliers will even do it under sterilized conditions. So they'll do some on radiated devices, they'll do some on ethylene oxide sterilized devices, or materials, I should say, not devices. And then they'll apply for the FDA to get a device master file. And what that means is now they have a file with the FDA with all this testing done under these circumstances, and you can even request that supplier to let you have access to that master file. And that allows you to now say, look, the FDA is aware of this, but more importantly, the testing, what testing was done. Now, that's important. You want to find out what testing is done. Just because they have a master file doesn't mean it's automatically going to help you, right? The master file will have either maybe just a cytotox test or maybe just even a discussion about the materials, all the way to complete testing on sterilized device. So it's still ambiguous on how well it would help you. But if they have it, definitely try to get that information from the supplier. Now, one of the biggest tools that we use to help evaluate the risk, and it goes back once again to the surface area ratio, is 10993-12. And this is table 1 out of 10993-12. And if you're not familiar with 10993-12, it's the sample preparation standard that us labs use to help prep the device for biocompatibility testing. And like I mentioned before, and you can see in the chart, it gives us ratios of either weight or surface area to volume. So the bigger surface area a device has, the more volume we can use to extract it. And that's important for us because we need that volume to test. Every test needs so much volume of extraction to run. So this helps us tell you how much sample we need based on how much volume we need to run the test and the ratio in the standard. Now, there's a nice little part of this right here, which is the plus or minus 10% for the ratio. And what that means for me as a lab is if you give me surface area ratio that lab A did, and we did a kind of a different, because we do these based off of paper calculations sometimes, and our paper calculation was a little bit different than the lab A's calculations. If it's within plus or minus 10%, then we can call those results comparable. And so what we look at this, though, is for changes, is if your change impacts less than 10% of that patient contacting surface area, then we can say that change is pretty minimal when it comes to the biocompatibility of the medical device. Now, I'm not saying that you can just pass that through, right? Because I just talked about before, dose makes the poison. So if that plus or minus 10% is still a very toxic compound, it doesn't matter, if it's plus or minus 10%, it's still going to be toxic. Also, if your device is huge, increasing 10% is still a big change when it comes to the patient. So I'm not saying this passes you by, but what I'm saying is this can be one input into your risk assessment to try to understand the impact of the change. You still have to look at the material, you still have to look at the processing, all those things we talked about to make sure you understand the risks of the patient. But this is one input that you can look at to try to evaluate the impact of that change. If it's less than 10%, then that means that your biocompatibility is probably not going to be as impacted. And so even retesting probably is not going to give you different results. Like I said, there are some exceptions to that, right? So that's just something to consider using a standard. Like I said, this is still in the standard right now, this table 1. It's a little bit different from that one, but it's still in there, the plus or minus 10%. Now, the one thing I do want to hit home, and it's something that has started to become much more used, just not to evaluate changes, but also just to look at biocompatibility. The new MDR is really kind of emphasized, and FDA has emphasized the desire or the benefit of chemical characterization. For me, I love chemistry for biocompatibility. I think it gives a lot of good information about compounds. It's not the save all of biocomp, right? There's still things that we need to do outside of chemistry, and chemistry has its flaws. It's not perfect. So, but one of the things that chemistry can be very useful for is evaluating changes, especially if you have a chemical evaluation from the initial device, and we have one of the new device. Now, I do want to tell you that one of the faults of chemistry is there are some inconsistencies in chemistry, and not just chemistry, but from devices. The chemical profile from lot to lot, or even devices ran at the beginning of the lot to the end of the lot, can be different, depending on what's exposed to during the manufacturing process. And the chemistry itself can have some variation, and not necessarily, hopefully not in the type of compounds, but the amounts can vary. And so this isn't like an FTIR that matches up and say, oh, we're identical to this, so we're good. What you're using this for is to understand if there's any differences in the number of compounds or the amount of compounds from initial to the change. And if there is, to look at those compounds and the amounts to understand the risk to the body. So it gives you quantitative data to be able to evaluate to help you understand your risks. So there is a lot of power that comes with chemical characterization evaluating changes. Now, I mentioned FTIR. Depending on, once again, this is all about risk and about how much testing you need to put in, if any, to help mitigate that risk. The first ones, the first chart I have over there is just simple tests based on low changes. Like if you are changing an HDPE portion of your device, a small portion of your device, to a new HDPE, something like FTIR, DSC can help gather information to show that that change has not been impactful. Those are very cheap and quick tests, and if the risk is low, these are things that can help mitigate those risks. This wouldn't be the right test to use if you're changing a complete supplier of like a silicone or durometer or something like that. We probably see differences here, and even that, if it's a high risk device and you're changing a lot of material, these will probably won't be sufficient to justify the change. But if you're changing, if your supplier says we're changing a polymer, but this new polymer is identical, you don't have to worry about it. Don't just take their word to it, right? Go in and do some of these simple evaluation tests to help test to understand if there is any differences between the old and the new. This can be done on the material itself. We do recommend that you go through processing before you do it, so that any impact from the processing can be put in there. Obviously, that depends on the material changing. If your device is going from an ASTM grade implantable stainless steel to a new supplier of the same ASTM grade, that's a low impact change. And so that's one that just an evaluation may be needed, no testing. So these are things to think about. Some of these tests, for example, metals. This is a great one if you are, ICP-MS looks at metals, chemistry test that looks at leachable metals. This is a great one to use if you are changing metals and you still want to just feel comfortable about that change. There's a lot of environmental labs around that can do this test for relatively quick and cheap, and you can get a good comparison of the leachable metal profile of your old metal and your new metal, just to help you understand if there is any impact. If there is a difference, that doesn't mean you have to repeat all your testing either. We can look at the different metal or the higher amounts of metal coming off and understanding from a toxicological standpoint, is there any increased risk from that change? We talk about some physical chemical tests down there. The other one that we use a lot to help evaluate potential change, this goes into unknowns, the unintended impacts, is that cytotox test. I talked about changing a location of the manufacturing plant. The cytotox helped us find that unintended impact. And so it's relatively inexpensive and quick. It's very sensitive. So it's a way to kind of help evaluate the differences in changes. I would run them together. I wouldn't run just a new cytotox on the new device and not rerun it on the old, because it's a sensitive test. Not all failures necessarily mean that it's an unsafe device or an unsafe change. We'd want to compare it to the predicate device to see if there's been an impact in safety from the change. Now, if you have a more aggressive change, let's say you're changing, a stabilizer or in a plastic, or maybe even going from one plastic to another type of plastic. This is where extractable leachables might be, the full-blown extractable leachables may be more useful. Or start looking at other tests that, depending on the use of your device, if it's skin contact or if it's very limited contact, maybe it makes more sense to rerun some of the traditional biocompatibility tests. So those are decisions that have to go in those risk assessments. So just to give you a sense of some of the breadth of our chemistry, right? So, these are all chemistry compounds can be broken down into two categories, organics and inorganics. Organics means they have carbon, and there are a series of chemistry techniques that we run to look for organic compounds. Those are ones like volatile organic compounds, semi-volatile, and non-volatile. The difference there is just the molecular weight of those compounds. So the heavier the molecular weight, the bigger the compound is, the more it goes towards non-volatile. And when it means non-volatile, it means it just doesn't evaporate very easily. It can sit on your, like lotion, for example, can sit on your desk, and it takes a long time, if ever, to really evaporate off. Where something like alcohol can sit there and evaporate, you can see it evaporate off. And so the alcohols are more volatile, and so they would be on the volatile organic compound screening, where the lotions and stuff like that would be down on the non-volatile. So those are the screens for organic compounds. And then for inorganic, it's metals, and that's where the ICP-MS comes in. So these, based on your change, we might be able to kind of identify a certain chemical technique instead of running a full-blown extractable leachable based on what's changing in your device. If you're adding a stabilizer, and we can look at the composition of that stabilizer, we may recommend a certain chemical technique, not the whole one, to evaluate the leachable profile of that stabilizer, and then the tox profile. So it could be something that's quicker and cheaper than running a full-blown extractable leachable based on the change itself. That's the benefit of evaluating a biocompatibility risk of a change instead of a brand new device, is you're focused. You're more focused on the change itself, on the risk of that change, instead of a lot of unknowns. So that's something to think about when you're looking at chemistry to help you on that. So this is just kind of how we look at the different chemical techniques to look at those volatiles and semi-volatiles. So like I said, if you're looking at inorganics, not carbon-based, it's metals and metal oxides. Any of the organics, once again, we're going into VOCs, S3OCs, and non-volatiles based on this molecular weight. So when we don't know what we're looking for, we open up our scans, we try to find everything we can. This is the full-blown extractable leachables that everybody's kind of dealing with right now in the industry. So those are helpful if the change is really not known or the impact is not known. We may have, and the device is a risky device, so we may have to go into more of a full-blown extractable leachable profile. But the endpoint of that full-blown is we get identifying peaks. So as you can see, there are peaks, and we can look at those compounds. The higher the peak, the more of it's there. This gives us the way to give semi-quantitative amounts of these compounds. And this is what you can overlap if you're doing two of these with it, like your traditional chemistry and your new chemistry. You can overlap these to try to understand the impact of the change. Now we're going to talk a little bit about cytotoxicity, because cytotox can be a useful tool to, especially when you're looking at those non-intended impacts of a change. It's a good screening thing to test just to see if there's an impact to a change you're not aware of. So for cytotoxicity, for those of you that I'm hoping, because it's one of my loves, I'm hoping that most of you guys are familiar with cytotox. If you're not, cytotox is a quick in vitro test. It's ran on cells. These cells are called L929 cells. They're mouse fibroblast cells. And so they're a adhering cell line, which means they attach. So they attach to the bottom of a plate. We extract your device in cell culture media, and we put that cell culture media after the extraction onto the cells. So any chemistry from your device is leached, just like I showed you before, but instead of identifying the chemistry like we do with extractables and leachables, that chemistry is now sitting on those cells and interacting with those cells. And anything toxic would now cause either the cells not to grow very well or cause them to die. And these are things we look for depending on the type of cytotox test you run. The one we're going to talk about today is the MEM elution. It's kind of the old qualitative test, but it's the quickest and cheapest test we have right now. And it's a good screening test for these types of changes. So it doesn't give you the quantitative information like an MTT or an XCT test would, but the MEM does give you a good screening, especially when you're comparing the old and new change. So the ME evolution, these are the dashboard for the test for your information. It gives you the sample amounts we need. The turnaround time, like you can see, it's a very quick test. It can be done very quickly. And so it depends on how needed these results can be. And then some of the problems that we see in cytotox historically, you're looking at natural rubbers like latex. That's our positive control. So if you're changing a latex, it's probably, if we're going to run this test, we're going to have to run dilutions because it's going to fail. We want to compare it to the old device like I talked about. But also a lot of problems we'll see is metals. So you'll see things like silver, copper, zinc, things like that will cause failures. So just to keep that in mind, I call cytotox your best friend and your worst enemy because it is so cheap and quick, but it's also incredibly sensitive. And not all the failures actually contribute to a true toxicity to the patient. So when you're doing this test, just keep that in mind. But as I mentioned before, this test is ran with MEM fluid. You can see that right here. That's the cell culture media. We extract and then put the extraction onto these cells. So the cells are at the bottom of these wells. And then we score it from a grade from 0 to 4. And it's just based on percentage of cells affected. In this particular test, a 0, 1, and 2 is considered passing. A 3 and 4 is considered failing. Although if you get a 2, you probably should be ready to try to understand why you got a 2, even though for the standard it's considered passing. These are what the cells look like. So this is a 0. This is a healthy, happy cell line. You can see a mosaic pattern on the bottom. They look really nice. This is a 4. And as you can see, I can probably teach everyone on this call how to score a 0 to 4 in an afternoon. That's pretty easy to do. What gets difficult if you start looking at a 2 or a 3, trying to get a sense of really what 50% looks like. So that's the downside of this test. But luckily, most of the time you get 0s and 4s. You don't get as many 2s and 3s and 1s. And a lot of the labs out there, including Nelson Labs, have very qualified people who've been doing this for decades, and so they get a good sense of what the cells look like. But that's the downfall of this test. And to give you a little bit of a sense what the cells look like when they're growing, this is the movie that I showed during the holidays to my family to help them feel like they're biocomp experts too. But this is just the cell line over 8 hours under microscopy, and you can see how they're growing and splitting. This means they're healthy and happy. So you can look at the cell line during real time to understand what happens to them and understand the potential toxicity. So if you want this to show to your own family during the holidays, just let me know. I'm happy to send it over to you. So the last stage of this before we get to questions is I want you guys to remember it's not just about the risk assessment and any testing you need to evaluate or mitigate that risk. You need to have some kind of report at the end, especially if you have to resubmit a 510K, because ultimately telling the story is the most important part. When I talk to FDA reviewers, they get so frustrated because a lot of times the engineers know these devices so well and the material so well that they just kind of dump a bunch of stuff on the reviewer, saying, okay, this is the change, the durometer, this is the testing we did, we're good. And it's better to kind of talk through the risks that you identified, the testing you did to mitigate the result or the risks, the results of those tests, and how you feel like the results make you feel confident to accept the change. So we call this the biological evaluation report. It could be a change justification, whatever you want to call it, but it's that story you have to walk through, because if not, they can get lost in the details. And let me tell you, I've been on so many FDA calls with simple changes that turned into huge fiascos because we didn't just simply walk through the change with them to help them understand that the change is low, the risk is low to the medical device. Up here, this is the example that they put in, the FDA put in to their guidance document on what a BER looks like. I prefer, and I'm biased, I know, but I prefer the way that we kind of lay it out. I feel like it's more of a story. This one doesn't really tell a story to me. This one kind of can walk through the risk, walk through how it's used. And then the conclusion is based on the results to summarize the devices, or based on the changes, or however you want to say it, the device meets the safety requirements of ISO 10993-1. So that's kind of some people miss that endpoint, but that's probably the most important part. Telling that story is essential to submitting to the FDA, especially, or any regulatory body, to be completely honest. Okay, so 45 minutes of listening to me just regurgitate information. Sorry about that, but I'm hoping that you got a good sense of some of the thought processes when it comes into evaluating a change for biocompatibility. But I really want to kind of hear some questions you guys have. I want to try to walk through those if you have it. If not, then everybody can go to lunch a little earlier on the West Coast. So, but yeah. So anyway, if there are any questions, Taylor, that we have out there?
We have questions. Surprisingly, no requests for that video you showed to show
Oh,
at
come on,
holiday
guys.
parties.
I promise you that they'll love it.
You still have time. You still have time to obtain that footage there. Lots of questions coming in. Feel free though to keep submitting them. We have a couple questions on the touch points, which I know you mentioned very early on in the presentation, so we'll have to take our brains back. But do we need to consider biocompatibility testing only for patient touch points, or does this include operator, so service engineer, clinician touch points as well?
Great question. So technically it does if those clinicians or the user of the device are not protected by gloves or other things. So if it's a home use device, for example, or a non-surgical device where the clinician can contact it with their skin, then yes, we still have to evaluate the potential impact to that user too. Now, once again, I know I mentioned the draft guidance that the FDA has about intact skin. A lot of times the users are touching them with their hands. Or maybe intact skin, so the risk is still pretty low. So I'm not saying you have to test, but that does have to be included in your risk assessment if they're not protected. Even if they are protected, I would still put a line in there that says the users are in a surgical suite, they're gloved, whatever, so they're not directly contacting the patient.
Yep, rationale is important there. And then in that same vein, would an evaluation be necessary for a change to a material of an internal component of that device that does not contact the patient at all?
Yeah, so that goes into that question about the indirect patient contact. So if it's completely sealed and there's no risk of having liquid going into that component and then back out to the patient, or during sterilization going into that and then going back into a portion of the medical device, then it's out of scope. Then the justification, you still need to assess it, and that's the assessment, is that it doesn't actually have either direct or indirect, possible indirect or direct patient contact. Now, if it's like a portion that could get some blood that comes in and blood comes out, and by the way, there's a difference between worst case and ridiculous case, right? So like, just everyone can think of a scenario where something could go in and something could come out, but we're talking about realistic worst cases here. Something where, yes, something could break or something and get exposed. So those are the considerations. But if it's not possible under realistic conditions, then there'd be outside a change. But you're still evaluating it. That's just your evaluation, is that's not going to be contacting the patient.
Gotcha. Okay, that makes sense. Bit of a longer question here. We have a 3D printed device and our raw material supplier shut down their operations. The contract manufacturer sourced a new material that they claim is the same-ish, in quotations there, to the original material. Supposedly the specs are the same. How do we know whether this change is acceptable? Do we need to redo all the biocomp testing that we supplied in the 510K, which was already cleared?
Yeah, so this is what I get every day, right? So I can tell you there's no such thing as same-ish from a supplier. And as much as I love them, to them it's like, oh, it's polypropylene, we're giving you another polypropylene, it's the same. And it might be from, like, when you look at what polypropylene means. But I think everybody who's a polymer scientist knows that polymers have so many additives and things that are different depending on, it could still be polypropylene, but there are going to be different components inside there. So what you have to do is it goes back to what we talked about at the beginning. The first thing you want to look at is the risk of the device. So it's a 3D printed device. A lot of 3D printed devices, or even if they're like classified maybe as low risk or even class 1, because they're 3D printed, the FDA sometimes has a little bit more concern about them because the process is so new. Depending on how you 3D print. Some of the 3D printing processes have been around longer and more known than others, but those are things you need to consider. And then based on how the device is used, then you have to go into the risk assessment for that change. Using some of those simple chemistry tests I talked about helps. So you could do an FTIR to actually compare the scan of the old and the new polymer to see how similar they are when they overlap, or DSC to look at their melting um, uh, uh, situations, or the information. So those are things you can look to try to just make sure that they're the same-ish, right? And that helps you identify and gives the FDA saying, we actually did some testing to help show that they're the same. If it's a high-risk device, and especially if it's 3D printed, then you might have to do more like chemistry, like the profile, to really understand how similar the extraction profiles are. So I know that's kind of a depends answer, which biocomp people love to give, but it does depend on how the device is used, the risk to the patient, and then the type of change in material. But those are some thoughts to go through.
Yep, definitely. Switching gears, we talked about FDA a lot, but would the EU notified bodies recognize the risk-based approach in the FDA draft guidance as state of the art?
Yes. Well, let me be careful here. We talked to
Yes,
TUSU. We
and
talked, yeah, it depends. We talked to a couple of the bigger notified bodies, and they are not necessarily saying, well, we're going to take that as state of the art, but the concepts in there are what concepts that are state of the art from the new 10993-1. So kind of the long answer is yes, the notified bodies are accepting this risk-based approach for changes. The only thing to be careful for with notified bodies is the quality of the person making the assessment plays a bigger role. And so the FDA will just take the quality of the assessments under consideration. If they agree with the science and it's well written, to them, that's kind of important, and not the title or the background and things like that. Although that's important, but it's more the quality of the assessment. For a notified body, and MHRA for England, they actually really want to see the person making the assessment is qualified to make those conclusions. So you have to send a CV usually with it, just to help, even if it's really well written, they want to make sure the person writing it has the qualifications needed to make those assessments.
And should you be proactive about
Yes.
sending that qualification, or wait until they ask? Definitely
No,
be proactive. Okay.
proactive. So every one of our assessments that we send to notified bodies, we will attach a CV, because
Okay.
we've just every single time we didn't, we got, hey, can we get your CV? So that's more and more what they've been asking for.
Okay. Okay. That makes sense. I really thought it was interesting about the location changes and the stories that came along there. So regarding the facility location changes, this person has a critical supplier of raw materials. They changed their location. Uh, this company initially performed the risk assessment, resulting in cytotoxicity testing. All the tests met the acceptance criteria. Do they need to perform any biocompatibility testing on the final product? It is an implant, although the raw materials are okay.
Yeah, so for me, this goes into discussing with your supplier. So if they all they did was change location and they kept all their machines, they didn't buy new machines or put new processing in, then just doing the raw material is probably fine. But if they're changing if they updated their machinery, or if they changed it or have new processes, you would want to do a cytotox after the processing, because you want to make sure that that hasn't changed either. But for the most part, location changes are pretty low risk. I gave you a worst case, but like I said, I've been doing this 20 years, and the vast majority of the location changes haven't changed the biocompatibility profile of the materials. But you just want to make sure that they've covered everything that could have changed with that location change.
Yep. Makes sense, makes sense. How does today's presentation relate to low risk class 1 devices?
Yeah, so for me, this isn't a difference between class 1, class 2. Well, I mean, that goes into your risk assessment, but everything goes through the same profile. So just because you're not submitting to the FDA because it's a 510K exempt, or it's class 1 in Europe, so you can self qualify, that's not things that I necessarily care about, because for me, it's about risk assessment to the patient. And I think any change should, at least, even if you're low risk, you should at least go through an evaluation of that change internally and have documentation on your rationale. Now, if it's low risk, if it's class 1 and low risk, then maybe testing isn't needed, and that's your conclusion. But you should still at least go through the same risk assessment to determine if that low risk is there.
Yeah, I think the FDA would call that documenting what you're doing and having a
Exactly.
rationale.
And remember, they can still audit you, right? So, I mean, you
Right.
still want to be able to show the thought process and rationale.
Exactly,
Sure.
exactly. And in this conversation, and just because I'm a quality person, I have to say in every conversation,
Yeah,
let's keep that
100%
mindset.
right.
Yeah.
100%. And that's why something like a good quality system like Greenlight could give you, right, could help you in those documentations.
Thor said it. I didn't say it. Keep in mind there. Here's a question about sterilization. Do you do the biocompatibility testing after or prior to sterilization of the product?
So the rule is a finished final device. So it would be after. But so once again, biocomps about depends. So obviously it's a process. So you have to look at the impact of that process to the material. So if you have a piece of titanium that gets EO, that's not going to change the titanium. If anything, it could make it better. It could remove something you might have on there as far as the humidity. So there are things that we can do to assess if you did biocomp pre-sterilization. But if you have a polymer that goes through radiation, there's no way we could evaluate the biocompatibility of that polymer without doing it after radiation, because the energy can have such an impact on cross-linking and things like that. So the rule is always do it after biocomp, but there are things that we can evaluate if biocomp was done, pre.
Okay. Okay. How do you establish correlations between the findings of chemical characterization and in vitro and in vivo biocompatibility testing?
Yeah, so there are some in vivo tests or endpoints. Let me just say endpoints, because we're working very hard to get away from as much animal testing as possible. So I'm just going to call them biological endpoints. There are some biological endpoints that have to be evaluated without chemistry. For example, implantation. The chemistry is about leachables. It's not about surface characteristics. And so hemocompatibility and implantation, things like that, irritation, even though there's some progress towards in vitro methods there, can't be done through sensitization or through chemistry, sorry. Sensitization is one that might be. So we're working on concepts in science and other industries that use chemistry to evaluate sensitization, but right now the FDA still requires a sensitization test to be ran. So there are ones that chemistry can't help you with. And so that's why but with a change, if you're overlapping, for example, if you only have one compound that's different, or maybe one compound that's higher, we could look at that compound and say, do we know that if that compound's a sensitizer? If it's not a sensitizer, then maybe it doesn't make sense to do sensitization. Is it an irritant, right? Is it toxicity? Is it systemic toxicity? And then you can look at the surface characteristics. Would the material surface characteristics be different? Do we have to repeat implant or not? So the reason chemistry is useful is it helps us identify a specific chemical to evaluate, because all the other unknowns and things like that might not have sensitization potential because you've already done the testing. Okay. So that's how we use it to help evaluate other biological endpoints.
Sure, sure. This person asked one question, but I think there's a question behind the question. So how valuable in your experience is a declaration of conformity or a statement from a supplier regarding absence of levels of substance of very high concern, of CMRs? They go on to say, if you don't have this information, what should you do? And I think the question behind the question is, if someone's giving you a declaration of conformity, what do you need to have on your side to back that DOC up?
Yeah, and actually there's even more to this question. I think I agree with you. There's a question behind the question, and then there's even another kind of information, which is with the MDR, right? It's the CMRs and the MDR. You're required to state about cohorts of concern, and you don't really know those unless you get those from your suppliers. And one, if you get those from your suppliers, you trust them. And two, if you don't, how do you deal with it? We've done webinars on dealing with CMRs that I can send over if you want to reach out to me, whoever asked that question, or anybody who's interested. I can send you a link to a webinar on how to deal with CMRs if you can't get from the supplier. But the first rule is get them from the supplier, because that's by far the easiest thing to do. And you're kind of stuck trusting the supplier. You can always do some of your own chemistry tests to see if you have like DHP there or some of the metals that are on the CMR list. But really, when it comes to the full list, the only way to know is to disintegrate or dissolve the material. And that's hard to do and from a chemistry standpoint. So if you can't get it, then we're stuck using chemistry. But remember, the CMR is about percent weight, and it's not about leachables. And so there's some kind of difference that we have to look at there. And that's a little bit tricky. So it's just so much easier to get it from the suppliers to submit to the notified bodies. The notified bodies just take the suppliers' declarations. So that's what you should do. But if you can't get those from the suppliers, reach out to me. I'll send you over the webinar that you can watch and get an idea of what your plans can be if you can't.
Yep. Yep. When there is a change in packaging material, does a separate evaluation for compliance to ASTM 2475 suffice to address the change? If not, what testing would be needed?
Yeah, so it could. I mean, that's definitely part of the risk assessment that we put in there, but it may not be the all you need. And most of the time, that's fine. Honestly, most of the time when you change a packaging, it's not a big deal because everybody uses pretty much the same packaging material. So that's what your assessment looks at: is are you using an insert that everyone else is used, or different packaging material that everyone uses? Those kind of things are easy. The hard part is when you start to add things in there that aren't known, and that's when you have to actually move more into the biocompatibility standpoint to look for bleachables. I talked about the label. We've seen ink leach through some of the Tyvek before, not Tyvek, sorry, some of the other materials before the paper getting onto the device, and so that can cause biocompatibility issues.
Gotcha. Gotcha. We may need a story hour here, but you mentioned not all cytotoxicity positive results mean true toxicity to the patient. Can you provide an example of when that may occur?
Yeah, so I'm on the TC194 working group for cytotoxicity, the ISO 293-5. I'm actually right now trying to put together an annex for that, for evaluating failures, because it's been a sore point in the industry where right now if you fail cytotox, it's difficult sometimes to justify that failure is not clinically relevant. A good example of that is silver. Silver is or copper, both are very cytotoxic, but not at very low levels, but are only toxic in the body at high levels. So you have this huge gap where you'll fail cytotox but not hurt a patient. So when you fail cytotoxicity, that's the key, is to try to determine the source. Look at the chemistry or look at the materials, talk to a biocomp expert, find out the source of that failure, and then look at the possible clinical impact. And if you can justify, especially through chemistry, to show that the chemistry is high enough to cause cytotoxicity, but low enough to not cause patient toxicity, then that's your justification, and that works quite well. The hard part is when you don't know where the failure is coming from, then it's hard to say it's not clinically relevant, right? So you really have to do some homework to find out where the failure is coming from.
Yep, understood. What are your views on carrying out in vitro biocompatibility testing on drug products, contact material for combination products? Do you think traditional pharma E&L should be sufficient?
Yeah, so there's a lot of answers to this one. So it kind of depends, one, on the branch of the FDA you're submitting to, right? CDER is different than CDRH when it comes to these types of evaluations. So depends on your device combination and the mechanism of use. That depends on which branch you go to. But let's say you're in CDRH, I would definitely recommend a pre-sub with the FDA on that one. The reason behind is because a lot of times our traditional biocomp tests aren't set up to deal with drugs, especially like cytotox or even some of the animal tests. This extraction ratio doesn't take into account drug doses, right? So there's a lot of specialized sample preparation that needs to go into those types. Plus, you got to look at the device itself, you got to look at the drug itself, and you got to look at them combined together. So those three endpoints all have to be evaluated. So work with a biocomp lab and then come up with a good test design to meet those endpoints that maybe aren't traditional biocomp, right? So traditional E&L may work for a lot of the endpoints, but definitely then do a pre-sub. With CDER, they're more used to seeing that type of evaluation, but for CDRH, we want to talk with them beforehand.
Gotcha. Yep. Always ask the questions first. I'm sure a lot of people are thinking this, but should these biocompatibility tests always be done by third parties or in-house? Or I can answer that question for you. You should always go to Nelson Labs for all of your testing needs. But what's your
take Taylor said it, I did it.
We're even now. We're even.
No, so I mean, I would love to have all the testing come to Nelson, right? I mean, that's perfectly fine. But to be completely honest, it depends on the quality. When it really comes down to it, if you have an internal lab that has high quality and their GLP biocompatibility tests have to be done under good laboratory practices. So if you have an internal lab that's GLP and independent, then that totally works. There's a lot of large companies out there that have internal labs that are GLP that do their own biocompatibility testing. The hard part is a lot of right now, and we're working on this, but right now there are animal tests that need to be done for biocomp, and it's hard sometimes to have an animal lab for a company. That's a lot of work. So it depends on the test. But some of the tests we talked about here, like FTIR, DSC, those kind of tests, for sure can be ran in a company. A lot of them have those instruments and can be ran. Just remember that you have to justify the testing to the FDA and be able to answer any questions. And so you won't get a whole lot of support for free from someone like us unless you do the testing with us. So that's the thing you have to think about.
Yep. And I'm assuming from your earlier point, the qualifications of the person doing the testing would come into play there as well.
Yeah, and the validation of those tests and everything.
And the validation.
Yeah, a lot of people have really good internal labs and they do great testing and the FDA accepts it. So it's not a mandatory thing. It just depends on how good you are.
Right, right. Stainless steel instruments. What is the consensus on finding for release and toxicity of metal ions and other elements within the alloys?
Yeah. So this is great. So the first thing to look at is trying to get that idea of the formulation of the metal from your supplier, right? So we talked about like ASTM grade or other types of grades for implantable stainless steels. You also want to look at the composition. This goes into CRM2 or the CMRs, excuse me, because for example, nickel is on there, and obviously stainless steel has nickel, so you have to try to understand how much of this nickel is in there. But then when you do the chemistry, so for instruments, I probably wouldn't do chemistry because it's a short-term contact with the body. The biocompatibility endpoints are like cytotoxicity, sensitization, irritation, acute systemic toxicity, and material-mediated pyrogenicity. Those tests are easier to run than running a full-blown chemistry. But if you are needing to look at the metal ions coming off, we can run an ICP-MS and look what comes off. And then we can look at the toxicity of those metals. So the one hard part about metals is they don't go out of the body very easily all the time. They tend to accumulate inside the body.
Yep.
So if it's short-term contact, no big deal. But implants, sometimes you have to consider that too. So those are the considerations we'll take into account if you want us to.
Okay, duly noted. I think this is an interesting one. Can it happen that the interaction of biocompatible materials become non-biocompatible as use goes on?
Yes, so there's a couple things I want to talk about here. There are possibility of synergistic effects. So you have a safe material for the size of it, right? And then you combine them together, and either they both have DHP, but they're safe by themselves, but you combine them together now, or two different types of plasticizers that have the same toxic endpoint. So as a toxicologist, you do try to look at synergies of compounds and how they could have an impact on the body. But that gets really the toxicological evaluation of mixtures is a discussion that at different toxicological meetings can blow people's minds away because the differences of that. So we put a lot of safety factors in our tox assessments to try to deal with mixtures. But when it comes to the life cycle of the device, this is something you definitely have to look at. So an implant can definitely become more toxic the longer it's in the body. If it degrades or breaks down, they could release bullets of materials months down the road that we won't see right away. So that's why when you're setting your biocompatibility profile or testing, you really have to look at the life cycle of that device. Can it break down? You have to make sure you look at the toxicity of that life cycle. So that's something you definitely want to work with a biocomp expert in to make sure your testing set up appropriately to evaluate that risk.
Yep.
And by the way, that's the same with shelf life too. If you may want to understand what happens on the shelf, it could get worse the longer it sits on the shelf, depending on the material.
How does this? We had a question about reusable medical devices. How does the fact that something's reusable pertain to this?
Yeah. So the two questions we always think about with reusable and biocompatibility is first, can it be used without being cleaned, right? So is the first use without being reused? And if it is, then all your biocompatibility needs to go through that first uncleaned device, because the actual process of reprocessing can remove leachables. It can remove detergents or things, residuals on there. So if it can't be used without clean, then we have to do that biocomp first. If it can't, then you want to do a reprocessing run and then test the biocompatibility. At the end of the reprocessing potential, so be it 100 cycles, whatever it is, we also want to reevaluate biocompatibility. That's looking at the material breakdown, like I talked about, or residual buildup, like cleaners being built up and not completely removed from the device. A lot of people use cytotox at the end of that, and that's in the standard, and that's something you can do. The other thing I'd love to do is actually look at the cleaners themselves and run different chemical techniques at the end of it to see if there's more chemistry at the end. And then we can look at a toxicological evaluation of those cleaners too. So there's different ways to do it, but there's definitely needs to be a biocomp evaluation at the end of the life cycle there.
Absolutely. Let's see here. Question on, so this company is using two previous FDA cleared devices and combining them. Will biocompatibility testing be needed with
So
this
that's kind
new
of,
combination?
yeah, that's kind of what I talked about was, adding different materials together. Most of the time, no, right? Well,
Yeah.
you still have to do evaluation and still have to understand the risks. But that type of thing, you usually don't. But you want to look and say, okay, you want to look at the materials and make sure you feel comfortable about those materials are well known, that you're not adding more and things like that by doing two together. But most of the time, if you have two approved devices and you're just adding together in one device, the risks are pretty low to the patient, unless you're changing the indication of use.
And would you leverage the test reports from the previous clearance of device A and device B as
Yes.
part of your own work? Okay.
Yeah, so you would use the report, the testing for device A and device B together, and then you would talk about the risks of combining them and say
Yep.
it's low based on the materials and processes.
Sure.
But one of the risks there, like if one has a cleaning process the same as the other one, now you're putting them together, they could be combining, right? So those are things you want to look at in your risk assessment.
Okay. Speaking of risk assessment, a question near and dear to my heart, what is the relationship between ISO 14971 and 10993-1? How do you show evidence of evaluating risk? Is it just putting it in your risk management file, or is it a test report, or maybe it's both? What do you think?
Yeah, and I'd love to hear your thoughts on this afterwards too, Taylor. But
Yeah.
for me, they actually play really close together. We reference
Yeah.
14971 and ISO 10993, right? Because the new version of 10993-1 is all about risk-based approach, and 14971 helps us understand that, right? So they do go together. There's even companies I know that takes the matrix of 14971 about the what we just talked about, the chance of the occurrence of the harm and the severity of the harm. They actually give a number for their biocompatibility endpoint. So there are different ways to do it, but I would definitely use both of those in your risk documentation and together as a tool. So Taylor, what are your thoughts?
Over documentation is always a passion of mine. Again, being a quality person, I am obviously keen to Greenlight Guru's way we have our risk management set up, and so including both, 10993 and of course 14971 and marrying the two. Maybe there's something you do with additional labels or tagging on risk items to say this specific mitigation or this specific hazardous situation is somehow related to biocompatibility, just so you can search through your big long matrix and see if asked how you're meeting the requirements of 1099.
Yeah, that's a shortcut. Good suggestion.
Yep. Thank you. You mentioned the regulatory trend is to use chemical characterization data in lieu of certain endpoint testing if appropriate. This person, Stephen, has heard that typically chemical characterization is not accepted for cytotox, sensitization, and irritation. Why is that?
Yeah, so you're right. So right now, if you look at the standard, the new 1093-18, it talks specifically about the endpoints, and not even that, the FDA guidance document also calls it out, right? So it talks about what endpoints you can use chemistry for and which ones you can't. And specifically, cytotox, sensitization, and irritation are not listed there. Now, the reason why irritation isn't is because irritation is a local response, and so it's not systemic. And right now, the FDA feels that chemistry can only be looked at as systemic responses. There are some thought, there are company or industries that use chemistry to look at irritants, right? So there are a list of chemicals that are known irritants, and if you see a chemical, you can look at that database and see if it's an irritant or a non-irritant. And we do that with changes, I mentioned it. So if you have one chemical difference in your ENL, you can look at the potential for that chemical to be an irritant based on how the device is used. But your full ENL for a new device, they won't take that. Sensitization is a little bit more of a hot argument within the standard committee because the pharmaceutical pharmaceutical industry does use chemistry to evaluate sensitization. They do have actually a threshold. It's 5 micrograms where anything below that's not a sensitizer. So, there are, and sensitization is systemic. It's local and systemic. It's a little bit weird, but it is systemic. So there is hope that one day we can convince, especially the FDA. We've been pretty good at convincing notified bodies that chemistry can be used for sensitization, but for the FDA, we're still working on that. My hope is that eventually we're also working on in vitro alternatives to both irritation and sensitization with the FDA right now. Dash 23 is coming out for the ISO standard, which is irritation, which uses an in vitro alternative. My hope is that one day we can run in vitro and use the chemistry to help support one another to get away from the animal tests.
Yep, very cool, very cool. We haven't said the S word yet, which is software. And software companies like to move fast. We like to be agile. Biocompatibility testing moves a little slow at times. It's
Snail
expensive,
pace.
snail pace. If this person has a couple things going on, they have software, they have firmware, they have machine learning. So there's a lot of changes happening to their device every week. I'm sure it feels like. Um, how can they approach biocompatibility testing in a way that is cost and time effective but still compliant?
Yeah, so we've actually helped companies like this to actually set up an internal procedure to look at changes that have lots of changes. And we put in, using thought process in 14971, we put in a biocompatibility specific protocol to deal with changes where anyone can just go through a workflow, a question and answer kind of process, and give them outputs to do. So based on this output, they do cytotoxicity. Based on this input, they do chemistry, whatever. And yes, the biocompatibility is slow, but cytotox is not, FTIR is not, right? So it's not all of biocomp slow. So it helps you identify what changes you can do quickly and what other ones are higher risk that you may need to budget a little bit more time. And so it helps with decision process too up front, looking at making changes, kind of understanding which ones are more impactful from a timeline standpoint. So that's one thing I would do. I would recommend setting up a procedure to help you understand internally the changes up front, which ones can be done quickly, and have anybody do it very quickly instead of going through a lengthy evaluation process.
Yeah. This person's asking on the topic of biological evaluation report, and I have my own follow-up question after this. How often should you be updating these reports? This person says that there's a new FDA guidance that just came out, but his biological evaluation report was made in occurrence due to 993-2018. Do we need to update every time a new revision comes out? What's your take on the update process?
Yeah, so let me say this. The official thing is when you're submitting to the FDA for a change, or you knew a CE mark is up and you need to go to the notified body, you have to make sure all your testing is to the current standards, right? I prefer to look at that from a yearly basis. I'd like to have every year someone look at their reports that they've done to see if there's any gaps. A good example of this is in 2012, we updated the irritation standard to include 3 rabbits. Well, if you did your irritation before that, if you're resubmitting a change to the FDA or getting a new CE mark, that you're no longer the current standard test, right? So there needs to be a gap analysis done. Doesn't mean you have to retest, just have to see if adding a 3rd rabbit would change the results. That's better to kind of have on top of things instead of like, oh no, we have to submit our CE mark, our rabbit irritation test is not up to date. We have to now retest that. Now we're 4 weeks out, right? So I prefer to go through that. You can even pay a company to do a gap analysis or whatever, just to make sure that the testing is to the current standard. And it doesn't mean the current year, because we don't change testing very often. So
right.
Even though the standard update doesn't mean the testing's changed. So it's really just to see if the current testing's still state of the art.
Absolutely. And my question is, again, as your resident crazy quality person here, what are some of the biggest mistakes that you see on these evaluation reports, and what can people do to avoid making those mistakes now rather than when someone's reviewing the file?
Yeah, the biggest mistake, and I mentioned in my presentation, is that they just dump data. So there's no flow to it. So and I'm telling you, I talked to a lot of FDA reviewers, and they get so frustrated when they just get a bunch of Adobe papers, right? Or even worse, when they get nailed in actual papers, and they're just like, this is the change, and these are testing, and there's no way for them to kind of build the thought process through it. And I think you said it right, over documentation. This is a story. You really have to hold the reviewer's hand through your thought process, through what you did, and why you did it, and at the end conclude that why you feel like it's safe. And that I've seen so many people go into problems, and the FDA is confused exactly what they changed and why they did this, and it ends up being a mess. So for me, it's really about making sure you're doing the right flow in your evaluations.
Absolutely. And this is true for, a CAPA report as well, or a complaint. It's if you handed it to someone who isn't aware of every aspect of your product and process, how can you write it so it makes sense? And I think biocompatibility testing and reporting is the exact same way.
Exact same way.
Let's structure it. Let's be clear and concise. Let's not, I guess, over documentation is one thing, but there's I forget what you said. There's crazy, and then there's like ridiculously
Ridiculous
crazy.
case, and then there's worst case and ridiculous case.
Yeah. So let's make sense of our data, knowing that we need to provide enough to tell our story, but not too much to overwhelm the poor person reading our document.
Exactly.
Um, when we were talking about in-house testing and of course Nelson Labs, Hans is asking if the device we are manufacturing is assembled of several different materials from different sub-suppliers, what can they do to not make shortcuts, but get ahead of performing the full evaluation of material themselves? How can they make their lives easier?
Yeah, and so this is something that we help clients with quite a bit. If they have even if they have a catalog of materials that use, or devices that use similar materials, or have devices with multiple materials that they can use throughout different devices, things like this, we can look at doing a combination of like a monster device test. So we don't need a functional device to test. We don't need a real device. We need a device that represents materials and processes. So we can come up with a worst case device using a worst case extraction conditions, and then that could be applied to devices that are using within that bookends. So we have a lot of people that have like a different accessories or different configurations, or have different materials, and we end up having a set of testing that can represent a bunch of different scenarios, so that now, as long as they fall within those scenarios, they don't have to repeat biocom tests. They can just reference the risk assessment up front. So that's a good way to do that.
Yeah, that's good advice. Good advice. Lindsay and others ask this, but we've mentioned it. Qualifications for risk assessors. Is there any specific thing you would want to see on someone's CV or resume? Any courses you can recommend?
Yeah, so every lab out there will do their own biocompatibility courses. We do. We're actually looking at doing our own certification for Nelson, so you can be a certified biocompatibility expert for Nelson. Yeah, so we're actually developing that training right now, just based on this. We're trying to work with Amy on that to try to make it more official. So there are things in the works. Unfortunately, right now, there's not a degree that really works well. I mean, like, for example, I have like 5 or 6 board certified toxicologists that I know, and they're on my staff. And most of them I wouldn't trust with the biocomp. I trust them all day long to assess chemistry, but not to do a full biocomp. And so it does right now rely on education and experience, mostly experience. But we're hoping to change that with industry certifications. And so that's the frustration. I've talked to quite a few really large companies with people who've been doing biocomp for a long time that are frustrated because they're getting pushed back by their notified body because their people don't have what they consider the true certification.
Right. I imagine it's similar to internal auditing or being an auditor, where there's courses you can take, but yeah, my major in college was not auditing. As much as I love it, it was not. So what can you do either through experience or shadowing or just time on the job to have yourself look qualified?
Unfortunately, that's right now until we get a certification thing worked out.
Yep.
Sorry, Lindsay and others.
Sorry, Lindsay and others, but back to the drawing board. A couple more questions here, and I'll wrap us up for today. You're doing great, Thor. I'm rapid fire going here, but
bring it on.
What should this person do when their supplier tells you he doesn't know if his materials or components contain nanomaterials? Just may not know.
So my first suggestion there is to not even mention nanomaterials,
Yeah.
right? So everything has nanomaterials. I mean, you think about the minute degradation of materials when they use the wear, how it's used in the body, it's going to be shedding things below 1 micron. And so everything can release nano. For us, nano is actually there for a purpose. If you're putting nano in the device for a particular purpose, to release, to attack cells, whatever it may be, then we go through the nano kind of category, the protocols, which is much different. It's kind of like the drugs. We have to test them completely different. We have to really look at them more like a drug than we do a medical device, even though they're materials. But just because your device could have nano on there or shed nano, for me, I would just stick with the regular biocomp unless you're intended to use nano or claim nano for a certain purpose. That's really where we would get down to that level.
Gotcha. Gotcha. Last official question here, and Thor, I'm sure you'll get an email of all of the questions that I wasn't able to throw at you today. This person has a a 510K cleared device that is mainly a colored fabric. Would you need to conduct a new biocompatibility if new colored fabric is used in the device? If so, would a new 510K need to be submitted? I think also we can make this more generally. Where should people go to help them decide if a new 510K is needed?
Yeah. Well, you can talk to experts that would help you, like Greenlight, right? They could help you know if 510K is needed. There's FDA also has a website that help you walk through when a new 510K. They have webinars they've done, things like that. When you start looking at the impact of the change from biocompatibility standpoint, I mentioned colorants. I'm not going to go back on that soapbox. But really, when you're changing colorants, one of the good things we can do is if you can actually get the formulation of the colorant from your supplier. So what's in there, every single compound, and the proportion of it, and then how much you're adding to it, we can look at that from a toxicological standpoint. And luckily, it sounds like it's intact skin, which is less concerning than something in the body. But that's all part of this risk assessment. We look at the colorant itself, the components of it, how much is used, where it's contacts the body, and then we decide if you need to do testing or not based on that change.
Awesome. Thor Rollins, an answer for everything. If people want to get a hold of you or want to enlist Nelson Labs in all of their testing needs, what should they do?
Yeah, so you can email me, LinkedIn, definitely LinkedIn with me. I love having LinkedIn. That's an easy way to get a hold of me. There's not many Thor Rollinses out there, so that should be pretty easy to
That's
find.
true.
My email is trollins@nelsonlabs.com, so you can always email me also, or just reach out to nelsonlabs.com, the website. There's places to ask people there, and you can always get in contact. So I'm happy to answer any questions you guys might have in the future.
That sounds great. Well, thank you all for your time today talking risk and biocompatibility with Thor Rollins from Nelson Labs. My name is Taylor Brown. I have been your moderator for today's session. Again, look for an email with the slides and the presentation, and be on the lookout for our next webinar and maybe even some in-person events later this year. So enjoy the rest of your day. Fingers crossed. Bye.